All right, thanks for coming tonight to this latest seminar in the Open Plant Pathology Seminar Series. I'm Adam Sparks. I'm an associate professor in the Center for Crop Health at the University of Southern Queensland. I'm a co-founder of Open Plant Pathology, which is an initiative that Emerson Del Ponte from Brazil and I started a couple years ago just to share the ideas around open science and reproducible research. And as a part of that during lockdown, Benjamin Schlesinger at ANU in Canberra came up with the idea to start doing a virtual seminar series. And our latest virtual seminar tonight is from Ms. Aurelie Quade, who is from the University of Southern Queensland in the Center for Crop Health, which is my home center. And Aurelie is going to talk to us tonight about the effectiveness and expression of resistance in maize to isolates of Puccinia sorghi from Eastern Australia. Aurelie, it's all yours.
Okay, thank you, Adam. So there we are. I think you've done a great job with the title. I might just move on then. So the first question you may ask is, where is Eastern Australia? And we are just down there. And so why is this study important? Well, we've got a great maize industry in Australia. However, maize common rust is becoming a problem. And we're going to be looking at ways where we can have some form and effective resistance. And that would be the objective of the study. How we do it will then lead us to a discussion. So why is it such a great industry? Well, the Australian maize at the moment is fetching about $60 a tonne. And we've got high grain quality in terms of no GMO status, low pesticide residue level, and we've got quite a good management of our environmental impact. Currently, only 6 to 7% is exported. That's about 80,000 tonnes per year. And because we've got such a close proximity to the Asian market, we have the potential to supply 14% of that Asian demand. And that's about $4.1 billion worth of shares in that market. The other thing we have to our advantage is that being in the Southern Hemisphere, we can supply grain to Asia when Europe, Russia, and America is not. So is it such a rosy story? Well, if it were, I wouldn't be here tonight, and it's not. Maize common rust, aka Puccinia sorghi, is a biotrophic disease, meaning that it requires a living host to get its nutrients from. It's an intercontinental pathogen. The spores are being picked up by the air current and dispersed over thousands of kilometers. And the yield reduction on susceptible varieties are about 40% and up to 50% yield loss has been recorded. Now, what you're looking at is the clonal stage of Puccinia sorghi, which is the uridinal spores. Puccinia sorghi has 5 spore stages, and we're only going to be focusing on that clonal stage, even though the sexual reproductive stage does occur in Australia. So why is it such an issue? Well, like a lot of rust species, it's got that ability to produce spores very rapidly. If you look there at 7 days post-inoculation, you can see the pustules are just starting to form. By the time we reach 13 days post-inoculation, that amount of spores on that tiny surface, if you take that back to the field level, you've got issues. And what's been happening in Australia is that over the last 5 years, growers and farmers have reported an increase in disease incidence and severity. The image you're looking at is a maize commercial line, and that's about a 6 out of 9 infection. A 1 being no disease and a 9 being completely covered. 6 is an unacceptable level of infection for a maize commercial line. So what do we do about that? Well, currently there's no chemical registered for maize common rust in Australia. Secondly, if there were, if you've got consistently high disease pressure, well, all chemistries are ineffective because you're going to have to do those applications several times, and that becomes uneconomical for farmers. The other thing we can be looking at is genetic, and you've got resistance Puccinia genes, so bear with me with RP gene, you're going to see that quite a lot. And those RP gene, most of them are located on the RP1 locus. Now, maize is naturally an outcrossing species. It requires that cross-pollination to keep that fitness. And what happens is that RP1 loci, they are very closely linked genes that are capable of frequent and unequal recombination at meiosis. And so what happens is that instability is generated with variation in gene copy numbers. Some lines can carry between 1 and 52 of those genes. They're not always expressed at once, and you can have those novel genes, combination or allelic variation. And often what happens is that you see that when you, in breeding, if you put two susceptible parents and you breed them together, well, the frequency of resistant progeny is quite high. So not super high, but high. The other thing I wanted to look at was physiological resistance, and that's adult plant resistance. As the tissue matures over time, you get that increased resistance in common rust. And so now we can gauge that physiological resistance versus the genetic resistance. How do we do that? Well, the objectives of the study were, firstly, I needed to know what was happening with those maize commercial lines. So the lines that are planted in different agroecological regions in Australia, or maize growing regions, what's the level of resistance? What do we start with? The other thing I wanted to know was that RP1D genes. RP1D remained effective in the US for over 35 years. And it's a very great gene for breeders to work with because it's easily introgressed into lines, maize lines that have got other desirable traits such as high yield, other disease resistance. The other thing we then did was, okay, if there's no resistance in the commercial lines, how about having other candidate resistant line? And we looked at RP genes, single and multiple, and again, that physiological resistance. So with the M&Ms, how does that work? Well, firstly, we had to get some isolates, and all those purple circles are showing you maize growing regions in Australia. And so farmers and agronomists and growers have been sending us rust samples. I then take those samples that have been sent from the field, I inoculate them onto a susceptible line, and I increase the spores and collect those spores as a sample collection. The second thing I do from that sample collection is I look at one pustule, I select that, and I increase the spores, and we get a single pustule isolate collection. And most of the experiments we did were from those single pustule isolates. Only one experiment was using the samples, and I'll explain why. And so now back on track, guys. The commercial line experiment, what did we do? We did 15, 7, 3. 15 isolates, 7 maize line with unknown resistance, and 3 sweet corn line carrying known resistance. Then when we looked at the RP resistance gene, we did 5, 21, 3. 5 isolates, 21 lines carrying known RP genes, and the 3 sweet corn line like before. And from that experiment, any candidate for resistance that was performing really well, that's the one we inoculated with the sample collection, where I bulked up most of my samples into a mega collection, and I inoculated all those lines with that mega bulk. And adult plant resistance, 110. 1 isolate, 10 planting dates. So I planted maize lines every week for 10 weeks and inoculated them all at once. And so to give you an idea, so really briefly, maize line, then named M1 to M7, and the resistance is unknown. And that was compared to sweet corn line carrying no resistance. That's our susceptible check. 1 resistance gene, RP1D, and 3 resistance gene, and this is our resistant check. The RP lines don't go into too much trying to read everything. All I want to point out is that we looked at no resistance gene, and that's your no RP line. And then we selected as many as we could get from that RP1 locus and one resistance gene. And then I thought, well, it's very well and truly great that we've got all those genes from the RP1 locus, but how do they stack up against maybe other single resistance gene? So now we've got other genes that are from other chromosome in maize. RPP9 highlighted in black, that's because this one is a resistance gene for Puccinia polysora. And I just thought it'd be nice to know what's going on and if it gives any protection against common rust. And down below, you've got your top performers, 2 resistance gene, 3 resistance gene, 4 resistance gene, just amazing, galore, resistance genes galore. And so how do we collect that data? Well, we looked at infection type. A 0 is no visible reaction. A node flake, you can see those necrotic flakes on the leaf with no pustule. Infection type 1 is those tiny pustules with quite a pronounced necrotic lesion all around. A score 2 is still quite small pustules, but as you can see, that necrotic lesion is not as pronounced. And then infection type 3 and 4, you've got quite large pustules, no necrotic lesions, and 4, the pustule become really elongated. So anything on this side of that yellow bar just there is an incompatible reaction type between the host and the pathogen, and anything that's a 3 and over is a compatible reaction type. The other data I collected was in percentage. So to give you a rough idea, 0% was no visible infection, 1% was a few dot pustules on the leaf, 50% is when you've got 50% of your leaf that's still available for the pathogen to colonize, and then 100% is completely covered with pustules and lesion, and there's no way you can fit in one more pustule in there. So what happened? Right, so bear with me with the charts. Each of those boxes is an isolate. On your x-axis, you've got your maize line and your sweet corn lines, and on your y-axis is the predicted infection. So there's 3 key messages to get out of this chart. The first one is that the maize commercial line and your 3rd dot before the end there are smashed repeatedly with all the isolates we used. And they were consistently as infected as the susceptible check. So you can see there, for every isolate, it's the same story. All those maize lines are performing really poorly. The second point of that graph is that RP1D just there, for instance, with isolate 24, is providing some protection. You know, you go from about 25% infection to around the 75% mark. That's a lovely drop in there. However, with RP1D for isolate 13, there's no difference between the line carrying that single resistance gene and the susceptible check and the maize commercial line. In that experiment, out of 14 isolates, 7 of them had compatible reaction type between RP1D and 7 of those isolates. And that's not a very good news. The third point is the line carrying the 3 resistance gene, your resistant check, is performing really, really well across all isolates. Good news, but move on. We need more genes. So now flip the chart around. Each of those boxes is a resistant line. And on your x-axis, you've got those 5 isolates. Those 5 isolates are quite important because I selected them for diversity. So the diversity is in terms of year of collection, location of collection, so Queensland versus Victoria, and also host between sweet corn and maize. And so the great news is down below there, we've got our candidate resistance. Yay! Fantastic. And they're performing really well. The other thing we have is that we've got a cloud of very susceptible line, but interestingly, stuck in the middle there, you've got lines that are showing differences based on isolates. And that in itself, for a researcher, is still a good news because we can use these as differential sets. One thing I have to mention as well is that all those lines, those candidate resistant line down there, when they were inoculated with that large bulk collection from all those different regions, they were still performing extremely well, and there was still no compatibility for any isolates collected between 2018 and 2020, and they are excellent candidates for resistance. When we look at physiological resistance, so if on your x-axis you've got your seedling age, by the time you collect the data at 14 days post-inoculation, those seedlings are between 4 weeks old and 13 weeks old. And straight away you notice that that group of seedling age there is not doing very well. If you average all of those data points, that's about a 70% average infection. And if you average the ones that are doing very well down there, that's about a 4.4% infection. That's a 93% drop in infection to common rust just on age alone. And you may think, well, that's wonderful. Well, no, it's not. Because even though you've got reduced infection, the infection type at that age is still an infection type 3. And those spores are likely to be carried out in the wind currents and reach fields that are yet lacking adult plant resistance. Now, the last thing we needed to determine, that is whether or not our population of rust is diverse. And the reason is for that is that if we've got high diversity within the pathogen population, what's the likelihood of those genes we're deploying to remain stable and over time? So that chart, all I want you to focus on is that just down there, you've got all your maize line and how many genes they carry. At the top there, you've got those really diverse isolates. All I want you to notice is that if we had a stable rust population, each of those lines there would have exactly the same color scheme, and they don't, which suggests that there is some variation. Now, let's change the subject a little bit. Is that now what you're looking at on that graph is on your x-axis, you've got days post-inoculation, so from 6 days to 16 days post-inoculation, and on your y-axis is your predicted infection. If we combine the data for all the RP lines for each of those isolates, well, there's not much difference there, is there? They're all behaving pretty much the same. You start quite low, close to 0% at 6 days post-inoculation. They're all about in the same mark percent by 16 days post-inoculation. However, if we look at the infection over time again, where we separate each of those isolates and we're looking at all those RP lines, there's quite a striking pattern. Isolate 24, 31, and 9, they've got that lovely way of sort of, you've got some resistant lines, some intermediate, and some very resistant. However, isolate 1 and isolate 73, I've got that ability, it seems, to be pulling the genetic apart between resistant line, susceptible line and resistant line. And there's only about, I think it's RP1K there, that sort of sits in the middle. And that's very interesting. So what's going on there? Sure enough, when you do your preliminary pathotyping, and I've just put the letter there with the lines I've used for those letters, the important things is that each of those isolates had different lettering. Even though the two last letters are the same for now, because the lines for letter 3 and 4 were all quite resistant, there is some diversity. None of those isolates had the same letters in the pathotyping. And so in conclusion, what I really wanted to point out was that we had those uridinial spores between 2018 and 2020. However, 2018 and 2019 were drought years. So the inoculum is there, just waiting for suitable weather conditions to come back. There was no resistance in the commercial line, meaning that those maize lines are likely to act as a source of spore production. And there could potentially be a huge conflict of interest with the maize and the sweet corn, because the sweet corn is a fresh produce and they really rely on the genetics to be able to supply that sweet corn to for human consumption in the supermarket, and that requires completely clean material. The other thing is that adult plant resistance should be used as an added protection to keep to maintain the longevity of the deployed resistance gene in environments of high disease pressure. We've got 8 candidate lines for resistance, and that's a fantastic news for breeding program. I just would like those lines to go through a bit more screening to determine whether or not that resistance or those resistance genes are likely to be durable or not. And the other point I would like to make is that all RP genes are valuable. And why is that? Like I was explaining to you, that ability of the RP1 locus to remingle and reshuffle those genes into new combinations means that even though some of those RP genes perform very poorly, they still are important to create those novel specificities. We now also have a differential set in Australia to monitor the common ROS population and to determine whether or not there are changes in the pathotype present. Now, the bad news is, I guess, is that some diversity has been found in the pathogen population. And the next part of my work is going to be to do a hybrid assembly between long-read pack bio from the first isolate I collected, and we can do that assembly with short reads. I've got 17 isolates that have done in Illumina on. And what we're hoping to do with that is not only to determine the extent of that diversity, but also to potentially pinpoint regions of the ROS genome that could be involved in virulence or could be involved in creating new specificities. And so I would like to acknowledge my dream supervisory team, and that's Professor Gavin Ash, Ben Stoddard, and Robert Park. I would like to thank the GRDC and USQ for the funding, and they've made that study possible, and also all the maize breeding and sweet corn companies down there. Without their help, I would never have had those maize lines coming from the US, and people from the statistical advice. So thank you very much, Guy. And finally, Peng and Yi, thank you so much for your help with the DNA work, and Matt, who's been helping me to just understand how to germinate the bloody spores. So thank you very much. Fire up if you have any questions, and back to you, Adam.
Thanks, Aurelie. Yeah, we do have a question already from Levente.
Okay.
How about similar studies overseas?
There has been, very good question. There's been one in South America done in 2016, which pinpointed that, yes, there was some diversity present there, and they also identified some RP genes that were suitable for their breeding program. But the good news is that the study we did was really worth doing, because some of the genes that they found that were susceptible were performing quite well in Australia and vice versa. So it was still worth conducting our study to understand the pathotypes that we had here and how those RP genes would do with the Australian population of maize rust. And there's been one in the US, extensive work in the US back in around 2000, and some work done also in South Africa.
I was wondering, because you kept referring to adult plant resistance.
Yeah.
And I know Queensland, we do crazy things. We plant at all times and whatnot. So does that really affect the severity of the rust outbreaks because we have these crazy planting schedules versus someplace like the Midwest in the US where I'm from, where, you know, everybody goes out and starts planting as soon as they can in April, and by August, September, they're wrapping up.
Yeah, no, it does. So very often you hear growers saying that if you put your crop in too late, so from about that past Christmas time, so there's a critical period where if you plant that crop too late, the growers do know that if there's a wet weather event, they're going to get absolutely smashed. So I think adult plant resistance was interesting to capture because it would make a great added strategy on top of your resistance gene. If you know the environment you're growing maize in is going to be smashed at that time. Just sort of scale back.
Yeah, because I was thinking that, you were referring to the seedling infection. So I was wondering how often seedling infection actually occurred, because, in the US, I know that I can't recall seeing it on seedlings really in the US. Usually you have to wait till midsummer when the corn is well over your head to actually see the infection.
Yeah, no, that's right. So I think you may not see that on those very young seedlings. It's just interesting to notice that, let's say if you were to combine maize with other crop, at sort of that, not that we do it, but now we're looking at other ways of farming that's more sustainable. And you think, when you've got your mixed crop, you get lower disease pressure. And if you were to do that, it would just still be a good little bit of information to know that, well, if you've got that closed up canopy or partially closed up, those seedlings are not going to do very well at all. But no, you're absolutely right. It was just really interesting to see that once you pass that critical stage, when your canopy closes up, even though that's when you think you're going to get more rust, well, actually that adult plant resistance does provide a little bit of protection.
That's interesting. And so I know it's not what you're looking at, but how much of an issue is polysora in Australia?
The growers see it mostly in northern Queensland in that really tropical climate. Yeah. There's a lady in Victoria who chases polysora on maize just in case, because if it's what do you call that? Biocontrol, and polysora is on the list for Victoria. It's never, but it means that she's been sending me some amazing common rust and chasing southern rust.
Oh, that's great. All right. Very good. So were there any other questions from the participants? If there aren't, I think we can thank Aurelie for a fantastic presentation. I greatly enjoyed that. Thank you for sharing that. That was fantastic.
Pleasure. Thanks, guys, for your time, and yeah, thanks for the comments, Adam.
Oh, thanks for your time, everyone, for attending, and thank you, Aurelie. And we will have this up on our YouTube channel probably before the end of the week, I would guess. So,
okay,
thanks
guys. Take care. Bye.